using Drosophila, the fruit fly, to study mitochondrial dysfunction and the identification of therapeutic treatments. And this is a compound screening project. So we're taking advantage of an endogenous mitochondrial mutation within the fly, and that phenocopies the disease state associated with mitochondrial impairment. And what we're able to do then is administer to the flies different test compounds and look for compounds that improve the animal's locomotor function and or longevity.
And this sounds extremely interesting. It tends to tell me that there's some translational opportunities here that might actually result in clinical aspects. Is that possible?
It's certainly possible. I mean, you need to simplify complex problems, and mitochondrial disease is a very complex problem. And but our way of simplifying it is to go into a fruit fly, and it preserves the animal's systems, the neuromuscular system. And we need a level, an organism at this level, in order to test compounds. And it's something that really complements research done in other cellular systems as well as larger animal systems.
Well, UMDF has prided itself in being able to provide bootstrapping grants for researchers such as yourself for these types of projects. Could you tell our audience and our membership a little bit about the level of importance bootstrapping grants are to a researcher such as yourself?
No, they're absolutely crucial. You know, the NIH certainly funds large projects, but in order to get NIH funding, especially in this climate, you need years worth of preliminary work in order to even be competitive for that kind of money. And these projects wouldn't even get off the ground if it weren't for the support of foundations and other private donors in order to make this work happen. One of the biggest problems with research is sustaining a research effort. Biomedical research these days is complex, and nothing gets done in a year or two, or sometimes even 10. And it's just, that's the level of complexity of the problems that we're addressing. And it's difficult, especially in this climate, to sustain NIH funding for that length of time. And having a source of income, a foundation or a private source that you can turn to sort of fill those gaps and keep the people in the lab working, motivated, interested in working on these projects, rather than aborting the project to work on another project just because it's funded, is absolutely key to keeping progress on the future. I understand that it might not be intuitive to many people that work outside of biomedical research, but when there's complex problems, and certainly mitochondrial disease is an extraordinarily complex problem, in order to study it, you need to reduce the system. We can't study it in humans. It's difficult to study it even in primates or mice. So the reduction that many people make is to reduce it down into a cellular form. That has advantages from a research perspective. You get a high degree of resolution in your assays. What we've done is we've reduced it down into the fruit fly model, and where we have a whole animal, we have an intact neuromuscular system, and we can study these system-level diseases within the systems of the fly.